If you're enjoying these videos, subscribe. Let YouTube know that I'm doing a good job by subscribing to the channel, plus you'll get notified when I have new videos coming out. And finally, if you're enjoying what I'm doing, give it a like, just click on that thumbs up, and that way YouTube knows that you like it, and they'll pass it along to other people that might enjoy my content as well. I really appreciate it. Thank you again. Let's get started.
Our first speaker is a retired university researcher from the National Institute of Molecular Biology and Biotechnology, University of the Philippines Los Baños, or Biotech UPLB. She pursued her Bachelor of Science in Agriculture, major in Soil Science, MS in Soil Science, and PhD in Soil Science in the University of the Philippines Los Baños. Her primary interests and specialization are in soil science, soil microbiology, and biochemistry. She is currently working on toxicological study and pilot testing of Nutrio Biofertilizer for improved production of sugarcane in regions 3 and 6. Without further ado, it is my privilege to introduce to you our first speaker, Dr. Virginia M. Padilla.
Good
Hi, Ma'am
morning.
G. Good morning.
Good morning. Good morning, everyone watching this webinar in celebration of the 16th National Biotechnology Week.
Ma'am, if I may ask, why did you choose sugarcane as your research focus?
As a research focus, because the country has a large area for sugarcane production, approximately 422,000 hectares with 62,000 farmers, and this requires a large amount of fertilizer, and we know that the fertilizer is very expensive right now. So I got interested with this problem in order to solve the high cost of fertilizer and also to protect the environment against hazardous effect of the chemical fertilizer. And besides, sugarcane is contributing large amount of money to our economy annually.
Thank you, Ma'am G. Let's keep going, as I'm sure that our participants are now very eager to learn more about your research on sugarcane. Go ahead, Ma'am G.
Thank you. Thank you, JN. Today, I will be presenting to you the topic on Nutrio Biofertilizer for improved production of sugarcane saccharum officinarum. Why go for Nutrio? This is because the problem with the current practice are the following. There is low national average yield of sugarcane in the Philippines, 60 ton cane per hectare only. The second one is the high cost of sugarcane production, such as inputs on fertilizer and many others. Soil deterioration, soil pollution, resulting to environmental hazards, and non-sustainability. So this is the product description of Nutrio. It is a bacteria-based foliar spray biofertilizer. It promotes nitrogen fixation. It is a plant growth promoting biofertilizer, and it is an organic fertilizer that acts as a supplement to chemical fertilizer. What are the benefits of using Nutrio? Number one, soil pollution and chemical runoffs are mitigated. Soil deterioration is prevented and gives way to long-term sustainability of land. It promotes healthier and greener plants. The yield and income are increased by 10 to 20% while costs are reduced. The resultant toxicity assay of Enterobacter sacchari S18 on duck embryo indicated that percent survival of the embryo ranges from 30 to 100%. And except for those eggs which have been injected with 5% phenol, the survival is only 30%, meaning it is very toxic. And all the other treatments are have high viability, meaning that there is no toxicity. The resultant toxicity assay of Enterobacter sacchari S18 on duck embryo indicated that percent survival of the embryo ranges from 30 to 100%. And except for those eggs which have been injected with 5% phenol, the survival is only 30%, meaning it is very toxic. And all the other treatments have high viability, meaning that there is no toxicity. The pilot test in region 3 is shown in this slide. The first graph, the ton cane per hectare, and given the fertilizer recommended rate in kilogram NPK per hectare, farmers' practice is 230-110-50. For 1/2 farmers' practice is 115-55-25. And those with farmers' practice alone and farmers' practice plus Nutrio, there is an additional phosphorus and potassium fertilizer, while the SRA recommended rate and the 1/2 fertilized 1/2 farmers' practice plus Nutrio, only native soil phosphorus and potassium are on the treatment. And the results show that 1/2 SRA recommended rate of fertilizer plus Nutrio is comparable with farmers' practice plus Nutrio and SRA recommended rate of fertilizer. And it is also 10% higher than farmers' practice alone without Nutrio. Cane yield in these treatments is still higher than the national average of 60 ton cane per hectare, ranging from 77 ton per hectare to 85 ton cane per hectare. The sugar yield, there was no significant differences among the treatments. So it will be more advantageous to use 1/2 farmers' practice plus Nutrio if it is not significantly different from other treatments, because there is a 50% reduction in the chemical fertilizer. The pilot test in Region 6, EB Magalona, Silay Negros Occidental, is shown in these two graphs. The first graph is cane yield in ton cane per hectare, where the recommended rate of fertilizer in kilogram NPK per hectare is R. Farmers' practice 234-69-180, and for 1/2 farmers' practice 117-34.5-90. The result indicated that farmers' practice plus Nutrio and 1/2 SRA recommended rate plus Nutrio produce a significantly heavier canes compared with farmers' practice alone and SRA recommended rate plus Nutrio. And farmers' practice plus Nutrio have an advantage of 31.5% heavier compared with farmers' practice, while 1/2 farmers' practice plus Nutrio 27.3% compared with farmers' practice alone without Nutrio. The sugar yield among the different treatments had no significant differences, and the yield increase of sugarcane fertilized with farmers' practice plus Nutrio is higher than 1/2 farmers' practice plus Nutrio, although the farmers' practice plus Nutrio is a little bit higher than the 1/2 farmers' practice. In 1/2 farmers' practice, there is a 50% reduction in chemical fertilizer. The demo trial in Region 3 is in Pando, Concepcion, Tarlac. The recommended rate of fertilization is the same as with SRA. Farmers in kilogram NPK per hectare, farmers' practice is 200, 200-60. 1/2 farmers' practice 100, 100-300. And the result indicated that cane yield of plants fertilized with 1/2 farmers' practice plus Nutrio is significantly higher by 39.5% compared with farmers' practice alone. And this is 43.1% higher compared with farmers' practice plus Nutrio. The increase in sugar yield in LKG 1/2 farmers' practice is 35.5% higher than farmers' practice alone, and 33.4% higher compared with farmers' practice plus Nutrio. This is the demo trial in Region 6, EB Magalona, Silay, Negros Occidental. The blue ones are the cane yield, ton cane per hectare, and the red one are the sugar yield in LKG per hectare. The results indicated that farmers' practice plus Nutrio have significantly higher, heavier cane compared with farmers' practice alone and one-half farmers' practice plus Nutrio, but no significant differences on the sugar yield. However, the yield advantage of farmers' practice plus Nutrio is 16.2% compared with farmers' practice alone without Nutrio, and 12.6% compared with one-half farmers' practice plus Nutrio. Although farmers' practice plus Nutrio have greater advantage over one-half farmers' practice, there was no reduction in odd inorganic fertilization rate, while one-half plus Nutrio had lower advantage, but with a corresponding 50% reduction of inorganic fertilization rate. This is the demo trial in Region 6, Licuan, Barotac, Nuevo, Iloilo. This is the first graph is the ton cane per hectare, the LKG per ton cane, and the sugar yield per hectare. The result indicated that farmers' practice and one-half farmers' practice plus Nutrio have cane yield advantage over farmers' practice alone, although they are not significantly different. The farmers' practice plus Nutrio and one-half farmers' practice have sugar yield advantage over farmers' practice alone without Nutrio. Demo trial in Region 6, Tiwi, Barotac, Nuevo, Iloilo, indicated that farmers' practice plus Nutrio is sprayed with Nutrio twice, yielded 77.7 hectare ton cane per hectare, which is 8.1% higher than farmers' practice sprayed once, which yielded 71.89 ton cane per hectare. The LKG per ton cane and the sugar yield per hectare indicated also that farmers' practice plus Nutrio sprayed twice gave significantly higher LKG ton cane and sugar yield in LKG per hectare sprayed with Nutrio once, resulting to a significantly higher sugar yield of 134.74 LKG per hectare, and this is 22.9% higher over the treatment on farmers' practice plus once. This is Mr. Celso Lahoy-Lahoy of Bukidnon, the first adopter of our Nutrio technology. In his experience in sugarcane production in 2017, he did not use Nutrio, and he obtained 69 ton cane per hectare with an equivalent of 124.2 LKG per hectare. And in 2018, when he applied Nutrio, he obtained an average of 95 ton cane per hectare with an equivalent of 211.85 LKG per hectare. This is the summary of comparative partial budget analysis on the pilot test of Nutrio for sugarcane in regions 3 and 6 in San Martin and in Silay, Negros Occidental. The result indicated that farmers' practice and one-half SRA plus Nutrio for both pilot tests indicated that Nutrio spraying with Nutrio have an advantage over that of the farmer's practice. And the net change in income indicated that in Concepcion, farmer's practice plus Nutrio obtained 10,355 pesos and per hectare, and one-half SRA plus Nutrio 6,890 per 0.66 per hectare. And this is only due to the effect of Nutrio. While in Negros Occidental, in Silay, farmers' practice obtained in farmers practice plus Nutrio, the increase in the net change in income of the farmer is 23,000, while one half farmers practice plus Nutrio is 20,784. So both of the treatments with spraying of Nutrio have positive performances. This slide shows the comparative partial budget analysis on the demo trial of Nutrio for sugarcane in regions 3 and 6. And the same observation was noted that addition of Nutrio improved the net change in income. In Pando, Concepcion, Tarlac, very high ang response for one half farmers practice plus Nutrio, obtaining a 72,755 pesos net income per hectare. While in Negros Occidental, one half farmers practice was the net change in income improved by 36,774.43, and also with farmers practice plus Nutrio, 31,655 pesos. In the case of Licuan, Barotac, Iloilo, the same with farmers practice plus Nutrio have higher net change in income per hectare compared with one half farmers practice plus Nutrio. But both of these is due to spraying of Nutrio. In the case of Tiwi, Barotac, Nuevo, the farmer that sprayed his sugarcane plants twice with farmers practice obtained a 30,665 peso net change in income per hectare. In summary, Enterobacter sacchari S18, a component of Nutrio, is non-toxic, hence safe for use as foliar spray biofertilizer. The pilot test in both regions 3 and 6, both farmers practice plus Nutrio and one half plus Nutrio, gave high cane and sugar yield, but not significantly different. Though the increase in yield of sugarcane plants fertilized with farmers practice plus Nutrio is higher than one half farmers practice plus Nutrio, there is a corresponding 50% reduction of inorganic chemical fertilizer. The demo trials conducted in the areas in regions 3 and 6 indicated also that cane and sugar yield of the plants fertilized with the different treatments with Nutrio showed positive performances. The comparative partial budget analysis from the pilot test and demo trials indicated that using Nutrio in combination with either farmers practice or one half farmers practice can improve the net income of the farmer. Although the main objective of the project was addressed by Nutrio in reducing the use of inorganic fertilizer in the trials, the contribution of phosphorus and potassium added, or the native soil P and K, in increased yields cannot be discounted. This is the overview on the benefits that can be obtained from Nutrio. Nutrio improved the net income with farmers practice plus Nutrio or one half farmers practice plus Nutrio, and decreases the amount of chemical fertilizer used by 50%, resulting to less chemical fertilizers, less production costs, increase in yield, and long-term land sustainability. And finally, increased customer revenue, as you have seen in the comparative partial budget analysis. In all of this, I would like to thank everyone for listening this presentation, and all those who have contributed in the success of this project. And of course, thank you very much for the funding support of PICARD, the OST, and the National Institute of Molecular Biology and Biotechnology in UPL.
Thank you so much for that presentation, Dr. Padilla. Once again, to our participants, you may leave your questions or comments in the comment section. Just write to whom your question is addressed as well as your affiliation before posting your question. We will be choosing some questions that will be asked to our speakers later, since we cannot attend to all of your questions at this time. Rest assured that we will answer your questions directly in the comment section if they will not be chosen during this session. Now, moving on, our second speaker is an assistant professor at the Institute of Biological Sciences, also from UPLB. She is a graduate of Master of Science in Microbiology, and her primary interests are microbiology and virology. She is currently working on bacteriophages, rapid detection of major viruses infecting native and hybrid orchids, and rice virus. Ladies and gentlemen, it is my pleasure to give you Professor Jaymee Encabo.
Hi, JN. Good
Hi, Ma'am
morning.
Jaymee. Ma'am, I have the same question for you. Why did you focus on vegetables in your
Okay, so
research?
vegetable production in the Philippines is affected by bacterial diseases, no, particularly soft rot. Now, soft rot has been a long-standing problem affecting small farmers, particularly, and it is actually responsible for significant losses pre- and post-harvest. To date, there's very limited information about soft rot-associated bacteria, and in fact, control methods are also limited. That's why the development of an effective control method is necessary. And one promising control is the use of bacteriophages, no, which are actually bacterial viruses that can be used to control plant diseases.
Thank you, Professor Jaymee, for highlighting the importance of such research on vegetables. I won't keep you for long as I am sure that all our participants are now very eager to hear from you.
Thank you,
Go ahead,
JN.
Ma'am
Good
Jaymee.
morning again to everyone who took their time to attend this webinar for the celebration of the National Biotechnology Week. So my presentation is about the biological control potential of bacteriophages against soft rot of vegetables.
Good morning. In the next few minutes, I will be sharing with you interesting findings from our research work that focuses on the biological control potential of bacteriophages against soft rot of vegetables. There are several problems that plague the vegetable industry in the Philippines. Now, chief among them is how to increase production and how to transport products without incurring too much wastage. In perishable crops such as vegetables, freshness, excellent quality, and longer shelf life are always desirable. However, soft rot has always been a perennial problem not only in the Philippines but also in the rest of Southeast Asia. Currently, effective measures for this disease is yet to be developed. Now, soft rot is caused by several bacteria, most commonly Pectobacterium carotoverum, previously called Erwinia carotovera, and by Dikea species. Soft rot occurs worldwide wherever fleshy tissues of vegetables are found, and the disease can be found on field, in transit and storage, or during marketing. It has been reported that around 15 to 30% of post-harvest losses are due to soft rot. So in this study, we identified and assessed the diversity of soft rot Pectobacteriaceae and their associated bacteriophages. We determined the lytic activity of phages against soft rot Pectobacteriaceae and evaluated the bioefficacy of selected lytic phages against pathogenic SRPs. So in this study, we included 5 high-value vegetables, namely lettuce, carrot, cabbage, Chinese cabbage, potato, and onion. Samples were collected from 60 collection sites from the 11 provinces that we visited. So we collected a total of 915 samples, which included symptomatic and asymptomatic tissues. So for our research strategy, we employed a combination of selective and differential media for the isolation and screening of SRPs, followed by pathogenicity testing, molecular characterization, and host range determination. Alongside, we also employed an isolation and purification strategy for bacteriophages that would involve spot tests and plaque assay, bacteriophage characterization, and lytic activity assessment. Now, for our bacterial culture collection, we have isolated a total of 1,268 bacterial isolates using the combination of CBP and D3 medium that allows us to screen and identify SRPs. So out of that 1,268 isolates, we were able to identify 348 putative SRPs. So this slide would show you the distribution of identified SRPs based on CVP and D3 screening per crop. So this would give you the idea that among the bacteria that we have in our collection, the SRPs only comprise a minority. So this would mean that aside from pathogenic SRPs, there are also non-SRPs that we have in our bacterial collection. So for the pathogenicity test, out of the 348 D3 positive bacterial isolates, we were able to observe pathogenicity of 163 isolates to their original host. So that's roughly around the 46.8%. No? So this slide would show you the relative proportion or the number of pathogenic isolates of the putative SRPs that we have identified in our collection. So this was followed by the detection of SRPs using genus-specific primers. No? So using Pectobacterium dicea-specific primers, we were able to identify 163 out of the 348 putative SRP isolates, and they were genotyped as Pectobacterium dicea species using PCR-based molecular markers. After which, we tried to identify the 163 SRPs using 16SR RNA method. No? So out of that 163 SRPs, we were able to sequence 101 of them using the primer pair by Weisberg et al. In 1991. And then based on sequencing results, we observed that P. aeroidearum was the most predominant species of Pectobacterium in our collection, no, which comprised around 85%. So phylogenetic analysis using 16SR RNA gene and host range assay revealed that the 16R RNA sequences of the Philippine SREs are closely related, no? Just like what you see in this figure. No? Most are similar to P. aeroidearum. No? So interestingly, there were also 3 unique sequences that were obtained from onion and cabbage isolates, and the host range assay revealed host specificity of the bacterial isolates, where you have individual isolates that can infect 2 to 8 crops, and then there are also around more than 50% of the tested isolates that were able to infect 4 or 5 crops. No? So in this analysis, we used 19 sequences of Pectobacterium dicea as reference strains, and then we used the 101 isolates that we have sequenced using the 16SR RNA gene. Now, we also tried to look into the genetic diversity of some of the Pectobacterium isolates that we have in our collection by rep-PCR fingerprinting. And then these results would show that ERIC and rep fingerprinting revealed significant diversity among P. aeroidearum isolates. No? As you can see in this slide, we have identified several P. aeroidearum isolates, but then interestingly, these isolates exhibited significant genetic variation. No? So you can also try to look into this dendrogram that we have generated that would show you that significant genetic diversity based on rep-PCR fingerprinting. Now, as far as the bacteriophage collection is concerned, we have a collection of 138 bacteriophages, and then we found out that there were around 110 phages that produce lytic clear plaques. So these are the ones which we characterize as lytic phages. In terms of phage source, majority of the phages were isolated from symptomatic tissues, and most of them were able to infect P. carotovorum. No? So there are around 69 phages infecting P. carotovorum, 35 infecting P. brasiliense, 24 P. aeroidearum, and 10 phages infecting P. cacticida. So just to show you some of the virion morphologies of selected phages in our collection, we observe that most of them belong to the family Myoviridae, characterized by that typical head and tail structure, although we also have some phages that belong to Podoviridae and Siphoviridae. Now, in terms of phage diversity, we observe that morphologically similar phages infecting same Pectobacterium species group together. So based on this analysis, we were able to generate 4 clusters. So you have cluster 1 with the super BHR phages, meaning broad host range phages. Cluster 2, which includes P. aeroidearum phages. Cluster 3 with P. carotovorum phages. And cluster 4 that would include P. brasiliense and P. carotovorum phages. So it appears that the groupings of isolates correlate with their host range and geographic location. No? So in terms of the characteristics of selected phages infecting P. carotovorum, for this 3 phages that we selected, we further characterized them, and we observed that the phages were stable at a temperature range of 30 to 50 degrees, pH level of 4 to 9, and they would have a latent period that would vary from 10 to 30 minutes. No? And then these phages were said to be narrow host range, being able to individually lyse only P. carotovorum. Now, we try to evaluate the lytic activity of these narrow host range phages, and then we observed that single infection treatments resulted in bacterial inactivation as early as 1 hour post-inoculation. No? So as you can see, phages effectively inhibited the growth of P. carotovorum in vitro. No? So interestingly, tissue slices inoculated with bacteria, treated with phages, protective effect was observed to range from 30 to 60%, no? Indicating a significant reduction in tissue maceration. No? So we also evaluated the efficacy of phage cocktails against P. carotovorum at low and high multiplicity of infection. So similarly, we observed bacterial inactivation in both treatments, and the lytic phages were found to be highly effective in semi-implanta assays against P. carotovorum at the low and high multiplicity of infection experiments. No? So another interesting observation that we have was the protective effect of these phages against P. carotovorum. So we tried to vary the time of phage application relative to bacterial inoculation, and then what we observed was that all these 3 phages were effective in preventing soft rot caused by P. carotovorum. But then interestingly, the inhibitory effect varied, no, at 3 hours post phage application, wherein you only have phage A3 and D1 being able to inhibit the growth of Pectobacterium carotovorum from 0 to 3 hours post bacterial inoculation. Now, another set of phages which we characterized were those that were found to infect P. brasiliense. No? So P. brasiliense phages were found to have broad lytic activity. They were able to lyse a number of P. brasiliense isolates, and the tissue slice assays also revealed significant reduction in tissue maceration when we have the phages inoculated in tissue slices infected with Pectobacterium brasiliense. Okay. And then another set of phages that we have in our collection would be what we call our BHR bacteriophages. So these are broad host range phages able to infect different species of Pectobacterium. So in this panel, you would see that we have at the moment around 5 to 6 broad host range bacteriophages that showed very good broad lytic activity, capable of inhibiting the growth of P. odoriferum, 4 isolates of Pectobacterium aeroidearum, and uh, P. cacticida and P. carotoverum as well. So at the moment, we were having ongoing screenhouse assay using this broad host range phages against selected P. aeroidearum isolates that were found to be pathogenic among high value vegetable crops. So hopefully we would be able to share the results of this efficacy evaluation soon as we have this assays completed. So in summary, we have collected a total of 1,268 soft threat associated bacteria from high-value vegetable crops, and 22.4% of them were screened as putative SRPs. Now, 46.8% of the putative SRPs were found pathogenic on its isolation host, and we identified P. aeroidearum as the predominant species, which constitutes 85% of the total species tested based on 16S rRNA sequencing. And we observed significant genetic variation among the identified SRP isolates in our collection, as revealed by rep-PCR fingerprinting. In addition, our collection of 110 lytic bacteriophages showed high specificity and lytic activity to P. carotoverum, P. brasiliense, P. aeroidearum, and P. cacticida. Now, phages infecting Pectobacterium with broad lytic activity were isolated, and the semi-implanta assays showed that lytic phages were highly effective against Pectobacterium species. That would be all. Thank you very much for listening, and a pleasant morning to everyone.
Thank you so much for that presentation, Ma'am Jaymee. All right, 2 presentations done and another 2 to go. We are now moving on to the first Q&A portion of this program. But before that, again, let me remind our participants to post your questions on the comment section of our Facebook Live, simultaneously streaming at both DOST Picard, DOST Philippines, and NBW's pages. Let me call on screen again Dr. Padilla and Professor Encabo to join me and answer the questions from our participants. Our first question is from Mr. Adrian Velasco, and his question is addressed to Ma'am G. Ma'am,
Okay.
tanong po niya ay, what motivated you to work on biofertilizers like Nutrio?
Thank you, JN, for that question. What motivated me to work on biofertilizers like Nutrio? Because the technology was developed in order to increase farm productivity, and as I have said earlier, we have a large area for sugarcane production, and the problem of environmental concerns, which cause extensive and detrimental effects of inorganic fertilizer. And besides, sugarcane contributes huge amount of money to the country annually. I hope I answered the question.
Thank you, Ma'am G. Our second question is for Ma'am Jaymee, and it is from Miss Fatima Grace Miranda. Ma'am, she would like to know what are the predominant soft rot bacteria of vegetables in the field and after harvest, and how about its diversity? Ma'am Jaymee?
Okay, thank you, JN, for your question. So to answer Ms. Miranda's query, no, so based on our results, what we observe is that Pectobacterium aeroidearum was the most predominant species in our collection. So based on sequencing analysis, around 85% of the SRPs in our collection was P. aroidearum. No? So most of the P. aroidearum that we have in our collection was also found to have a broad host range, meaning majority of them were able to infect around 2 to 8 crops. No? So in terms of diversity, rep fingerprinting results showed significant genetic variation among these P. aroidearum isolates. Thank you.
Thank you, Ma'am Jaymee. Our third question is for Ma'am G.
Okay.
And the question is from Miss Maria Rizalin Ignacio. Ma'am, ang tanong po niya ay, can Nutrio be used for other crops aside from sugarcane?
Yes, thank you for that question, JM. Nutrio, yes, Nutrio can be used for other crops as based on the experience of farmers who got interested to test this Nutrio on crops such as rice, corn, vegetables, papaya, jackfruit, gayabano, and rambutan, and other vegetables. Okay. Thank you, JM.
Thank you, Ma'am G. Our next question is from Mr. Reynante Franco, and it is addressed to Ma'am Jaymee. Ma'am, he would like to know what experimental techniques can be used for ease of isolation and identification of soft rot Pectobacteriaceae.
Okay, thank you, JN, for that question. So, based on our research strategy, we utilize a combination of conventional and the molecular methods. So, for the conventional methods, we employed the combination of selective and differential media. So, just to recall, one particular medium that allowed us to screen for pectolytic bacteria would be CVP medium. So using this particular culture medium, easily you can screen out for pectolytic bacteria based on cavity formation. No? So once you grow those bacteria in CVP, you just look for pit formation, and then those that would form cavities in CVP were further screened using another medium, which is D3. So this time, the principle involved is D3 medium would allow differentiation between SRPs and non-SRPs. No? So based on sugar utilization. So parang may color reaction po kayong makikita doon. So kapag makita nyo na green to red color formation surrounding the colony, so that would tell you you were able to screen for putative SRPs. And then culture media screening was followed by molecular identification. No? So we used Pectodicea specific primers. So it's good in a way that by PCR you can readily identify which among the isolates can be genotyped as Pectobacterium or Dicea. And then, of course, very handy to validate the PCR-based identification would be 16S sequencing. So basically, that's what we did to readily identify SRPs from our collection. Thank you.
Thank you, Ma'am Jaymee. Our next question is for Ma'am G, and it is from Ms. Marites Manalo. Ma'am, she would like to know, when did you start developing Nutrio?
Nutrio was developed starting from September 2012 up to 2015, and we have another phase, this toxicological study and pilot test in regions 3 and 6, which just finished this year. Thank you, JN.
Thank you, Ma'am G. Our next question is for Ma'am Jaymee, and it is from Ms. Laarni May Castillo. Ma'am, her question is, what are the factors that might affect the stability of your isolated phages if you applied it in different condition or environment?
Okay, thank you for that very good question. No? At this point, results of our laboratory experiments are really quite interesting and shows promise. But then so far, if you look into the possible application of bacteriophages in field conditions, there's still a lot of factors that could affect phage stability. One important limiting factor would be exposure to UV irradiation. No? So in the field, it's possible that phages upon exposure to UV can result to inactivation, and then this results to a decrease in their lytic efficacy. So looking ahead, we are actually hoping that we can push through with the stability assays to determine resistance of our phages, particularly to external conditions or environmental factors, primarily UV exposure, exposure to various temperature conditions, and more importantly, the survival of phages in the phylosphere and the rhizosphere, because this would have important implications towards the mode of application if we are to formulate a phage-based product against bacterial soft rot. Thank you.
Thank you, Ma'am Jaymee. Our next question is for Ma'am G, and it is from Miss Sunshine Jane Kolkol. Ma'am, ang tanong po niya ay saan po available ang Nutrio for purchase at available na po ba siya commercially?
Thank you for that question. That is very interesting for our company that produced. We are the one producing the product Nutrio commercially. However, right at this moment, the product is directly ordered from us, because we do not have yet the distributor, and this is not yet available in the market. So only direct order from us by contacting us to our cell phone number 0917-1798-542. So the orders we receive, and then we ship it to the area, and they shoulder the shipping cost of the product. Thank you for that question. I hope answers
Thank you, Ma'am
your
G.
query.
Our next question is from Mr. Albert Malinaw Padilla, and it is for Ma'am Jaymee. Ma'am, he wants to know, how does biotechnology affect the propagation of agricultural products? Is the bacteriophage environment friendly?
Okay, to answer that question, looking ahead, if you talk about phage biocontrol, it really shows a lot of promise in terms of controlling bacterial infections. Now, hopefully, phage-based control would be able to more or less provide an alternative solution to the long-standing problem of bacterial soft rot in vegetable production. Now, one important feature of bacteriophages is their high specificity and environmentally friendly. No? So when you talk about bacteriophages, they particularly infect species of bacteria, which makes them very safe to use. In fact, there are already commercially available phage-based products in other countries, and these are generally regarded as safe to use already. So phage biocontrol is really a promising solution to control bacterial diseases. And of course, we hope that this would more or less help farmers, particularly those severely affected by the problem of soft rot, in order to reduce the significant impact of soft rot in vegetable production. Thank you.
Thank you, Ma'am Jaymee. So our next question is for Ma'am G, and it is from Mr. M. Lacto of Green Zymo. Ma'am, he wants to know, does Nutrio Biofertilizer affect the purity of sugar per ton cane value of sugarcane?
Per ton. How, in fact, based on the results obtained, the LKG per hectare for both farmers practice plus Nutrio and 1/2 farmers practice plus Nutrio, the LKG per hectare increased, and it also increased the net income of the farmer. Thank you for that question, Jaymee.
Thank you, Ma'am G.
Thank you, ma'am.
Okay, we are now on to the last question. The question is from Mr. M. Lacto again from Green Zymo. And it is addressed for Ma'am Jaymee. Ma'am, this Pectobacteria acts as biopesticide and also resides in the soil?
Okay, so to answer that question, our work focus on basic studies that aim to identify soft rot associated bacteria in vegetables. No, so the pathogen that we studied was Pectobacterium, and then what we wanted to determine was the efficacy of bacteriophages against Pectobacterium species. No? So in general, Pectobacterium species are widespread in the field, and they're easily disseminated through a lot of ways, particularly if you have like rotting vegetables left in the field. So this can easily cause contamination of vegetable crops in particular growing areas. So what we did was to identify the major soft rot Pectobacterium associated with vegetables, and then looked into the potential of bacterial viruses as biocontrol agents against Pectobacterium. So I hope I was able to answer that question. No, thank you.
Thank you everyone for sending your questions. I hope that you were able to get valuable learnings from our first two speakers, as they had presented two interesting technologies that will surely help our farmers. Now, before we move on to the next two topics, let me ask Dr. Padilla and Mom Jaymee if they would want to leave some words for our participants. Let us start with Dr. Padilla.
To our participants, thank you very much for watching and listening to the presentation on Nutrio as biofertilizer for sugarcane, improved production of sugarcane Saccharum officinarum. I hope I imparted to you some informations about this technology and the availability of this product in the future.
Jaymee?
Thank you to all of the participants for allowing us to share with you our important insights and findings regarding our work on bacteriophages. So overall, results of our work demonstrated the potential of bacteriophages to control soft rot disease of bacteria, although more research on phage biocontrol is necessary, particularly under field conditions. Our research have indicated that phage biocontrol has the potential to reduce the economic impact of soft rot in vegetable production. So we hope that you learned a lot from our sharing this morning, and thank you for sharing your precious time with us. Thank you.
There you have it, ladies and gentlemen. Should you have any questions or concerns that you want to further discuss with our speakers, you may reach them through their email flashed on your screen. I hope you are still with us. Again, this is the 16th National Biotechnology Week webinar on S&T Agri-Aqua Biotech applications for the improvement of the environment and economy. We now have a total of 500 plus viewers on Facebook. Thank you for joining us, and keep on sharing our live streaming link here on Facebook so that we can reach more viewers and further increase the awareness of the public on the positive impact of biotechnology, specifically on the agriculture and aquatic sectors. We are now in the second half of our webinar, and we're down to our last two speakers. I would like to remind our participants to leave your questions in the comment section in this FB Live post. We will try our best to answer your questions and ask them to our last two speakers during the Q&A portion. Before the end of this webinar, we will be flashing the link to our Customer Satisfaction Feedback Form, or CSF. All participants who accomplish the CSF will be issued with an e-certificate. Only those who submitted their CSF will be given the certificate, so please make sure to accomplish the form after this webinar. Likewise, the link will be posted in the comment section after the live broadcast. Those who are not able to watch this webinar live may still submit the CSF until 12 midnight today to be entitled for an e-certificate. Please give us 10 working days to process the issuance and distribution of the e-certificate since we have a lot of participants in today's webinar. In accomplishing the CSF, kindly make sure that you input correctly your complete name as you would want them indicated in the certificate. Likewise, please make sure that you input your email address correctly as we will be sending out your e-certificate via your registered email. Again, the link will be posted later after the closing program. All right, I will not delay this any further. Our third speaker is a professor at the University of the Philippines, Mindanao. He finished his PhD in marine science in the University of the Philippines, Diliman, and his primary interests focus on diversity and ecology of reef fishes. He is currently working on DNA barcoding and genetic diversity of selected marine fishes in Davao and Sulu Archipelago and along the north bifurcation of the North Equatorial Current. To discuss the fish barcoding and biodiversity database initiative in Mindanao, we are all very pleased to have Dr. Cleto Nanola Jr. Hi, Dr. Nanola. Good morning, sir.
Good morning, JN.
Sir, can you share with us the uniqueness of our biodiversity of fishes as compared with the other countries?
As far as the record is concerned, we have the highest marine biodiversity of reef fishes globally. So that is our strength.
Thank you, sir, for enlightening us. Our viewers will know more about this research in your presentation. Take it away, sir.
Thank you, JN. So once again, my topic is all about my ongoing project on DNA barcoding of reef fishes along the north bifurcation of the North Equatorial Current. Once again, thank you.
Hi everyone, I'm Ting Nanola, the head of the Coral Reef Resiliency and Ecology Studies Laboratory.
makes millions of copies of our target marker. This reaction is made possible by a thermal cycler and, of course, PCR kits. After PCR, we check amplification success using agarose gel electrophoresis. We allow the PCR products to migrate through an agarose gel using electrical charges. Electrophoresis resolves DNA fragments on the basis of their molecular weights. Smaller DNA fragments migrate faster than the larger ones. Using a reference molecular ladder, we can determine the fragment size of our PCR products. After electrophoresis, we view the gel using a gel documentation system. The presence of a bright band with an approximate molecular weight of 650 base pairs tells us that Q1 amplification is successful. We then send the PCR products to a biotech facility, such as the Philippine Genome Center for gene sequencing. Once we receive the sequence data, we prepare them for querying on online sequence databases. The resulting hits from the search serve as the genetic ID of our samples. We then compare this with the morphological ID. Aside from market surveys and laboratory works, we also conduct training workshops for our partner higher education institutions in Northern Luzon and Southern Mindanao. We have also provided internship programs on fish barcoding to senior high school students of the Philippine Science High School in Caraga and Southern Mindanao. We are currently preparing a laboratory manual which details our work. These are the same techniques that we introduce to our partner institutions so that researchers are fully guided and involved.
Currently, we have recorded over 150 fishes from wet markets in Northern Luzon. In Southern Mindanao, we have recorded a total of 373 unique species. If we go further the island chain of Basulta or Basilan Sulu Tawi-Tawi provinces contributes most to this number, with over 304 unique species. This means that overall, the reef fish diversity in Basulta is very high compared to our other study areas. Altogether, the DNA barcoding project has generated around 200 DNA barcodes. This data will be stored in the biodiversity database, which will contain all the biological information we have collected in the last 2 years.
I am Leo Manuel Estanya. Currently, I am an assistant professor of the Department of Mathematics, Physics, and Computer Science of the University of the Philippines, Mindanao, and also the co-proponent of the proposal research program Abraham. I am the one in charge in the creation of the database as repository of the morphological and molecular data that are gathered by the different projects in Mindanao related to biodiversity in general. The database also involves some data analysis tools which will be of use to the different researchers. It also involves spatial mapping of the different species which can be of use to the different government and non-government agencies for their planning activities involving biodiversity protection.
From combining the morphological and molecular perspective in biological specimens to creating a database that will be made accessible for all, we believe that bringing and developing these tools contribute to the research advancement we all envision for Mindanao. Mindanao is teeming with marine biodiversity. This was supported by the paper of Richard Mualil and colleagues, including myself, declaring that Tawi-Tawi is the heart of the Coral Triangle. It is undeniably a perfect opportunity to document the diversity of reef fishes, particularly in southern Mindanao, before they will be extirpated. Our unifying vision is to gather the local experts in the region and to include other stakeholders in order to sustain the marine biodiversity in the area. As an example is our proposed Abraham program that stands for Applied Biodiversity Research for Holistic Advancement in Mindanao, which will focus on studying our marine biodiversity at the species, population, and community levels. As mentioned, an integrative taxonomic approach combining traditional and contemporary molecular technologies will be utilized in studying biodiversity. It will take time for our goals to be fully realized, but we're taking the initial steps.
Thank you so much for sharing with us your expertise, Dr. Nanola. Now let's move on to our fourth and final speaker, who will share with us this morning the new micro propagation technology on queen pineapple. Our fourth speaker is a professor of the Visayas State University. She pursued her PhD in horticulture at the University of the Philippines, Los Baños, and her primary interests are plant breeding, tissue culture, and plant genetic resources. She is currently working on abaca and tissue culture derived planting materials of selected queen pineapple via somatic embryogenesis. Ladies and gentlemen, it is my privilege to give you our fourth speaker, Dr. Luz Moreno.
Hi,
Hi,
GN.
Dr.
Good
Moreno.
morning, GN, and also to all our listeners to this webinar.
Ma'am, may we ask your favorite pineapple
Of course, queen
variety?
pineapple.
Thank you, Dr. Moreno. I hope all our viewers have also tried the queen pineapple. And I know our viewers will know more about pineapple and your research in your presentation. Go ahead, ma'am.
Okay, so first I would like to thank for Picard for this privilege to share with you this technology that we developed. So queen pineapple is a very special variety which is very popular and in demand for its sweet and crunchy eating quality. And it is one of the most promising economic crops is commercially grown in regions 5 and 8, particularly in Ormoc City and in Camarines Norte. However, the availability of quality planting materials is highly seasonal and limited. So to help farmers and commercial queen pineapple growers on the problem of the availability of quality planting materials, we developed this somatic embryogenesis technology in the mass production of queen pineapple planting materials. So this morning, I will gladly share with you this technology. Thank you.
Good morning. Before presenting and discussing the technology that we have developed,
I would like first to thank the OSTP card for giving us the research funds and of course for the support and trust to conduct this research and this rare opportunity to present this technology on the occasion of the 16th National Biotechnology Week celebration in collaboration with the Department of Environment and Natural Resources. Now, the technology on the mass production of queen pineapple planting materials through somatic embryogenesis that I am going to share with you this morning is the product of the research titled Production of Tissue Culture Derived Planting Materials of Selected Queen Pineapple via Somatic Embryogenesis. Queen pineapple is a resilient crop best suited in typhoon-prone areas like regions 5 and 8. Now, this is the crunchy and very sweet variety preferred by the local market for fresh consumption with an increasing demand of 4 to 7%. The availability of good quality planting material is one of the most important elements of a successful agricultural production. Hence, the importance of healthy, vigorous, disease-free, uniform, and true-to-type planting materials. Pineapple can be propagated either from suckers, crowns, shoots, and slips, but the most commonly used planting materials are suckers, which can be obtained 21 to 22 months after planting or 3 to 4 months after harvest, with only 10 to 15 suckers per plant. Micropropagation of pineapple plants has many advantages over conventional methods of vegetative propagation. Now, it is more efficient, relatively cheaper, and rapid multiplication of quality planting materials can be attained in a shorter period of time. In the production of quality planting materials through tissue culture, selection of mother plants or explants is very important. So we sourced our mother plants directly from one of the biggest pineapple plantations in Ormoc City and selected the bigger fruits, and developed our sterilization protocol in the preparation of the explants. Using a series of soaking and washing with 70% ethanol, then 5% to 10% sodium hypochlorite solution, and rinsing with distilled water prior to inoculation, the rate of contamination was significantly reduced to only 5 to 10%. Direct organogenesis was done first using a protocol that we developed and optimized consisting of MS as basal medium supplemented with BAP and IAA growth hormones. And after 2 weeks in culture, the tissues started to develop and turned green after 4 weeks. And 8 to 24 weeks after inoculation, a number of multiple shoots have developed. The next step is callus induction. Shoot tips and leaf segments from 8-week-old cultures were used as explants for callus induction and grown in MS basal medium supplemented with TOFOR-D and picloram. The developed calli were again transferred into regeneration medium, and a series of 4 to 5 micropropagation cycles at 3 to 4 weeks interval followed. And the cultures were transferred to rotting medium and later subjected to acclimatization under room temperature for 2 weeks prior to ex vitro establishment. A protocol for ex vitro establishment was developed and optimized, which gives us 95% to 100% survival of seedlings in the screenhouse. After 2 to 3 months in the nursery, the seedlings were ready for field planting. The entire process of mass propagating queen pineapple through somatic embryogenesis until it is ready for field planting is about 12 to 13 months with around 8,000 to 10,000 plantlets or seedlings produced per crown shoot tip compared to the traditional sucker with only 10 to 15 suckers per plant after 21 to 22 months from planting. One of the most important advantages of somatic embryo-derived planting material is the well-developed and established root system compared to suckers. Results of the field evaluation on the performance of queen pineapple using different types of planting materials revealed that plants derived from somatic embryos produced more number of leaves in both Camarines Norte and Leyte areas under low and high density planting scheme. The somatic embryo-derived plants grows faster and even taller than those derived from suckers after 8 months from planting, even if the sucker planting materials were relatively bigger and taller compared to both the somatic embryo-derived and direct multiple organogenesis tissue-cultured plantlets, and significantly produced more suckers. After 3 months from harvest, the somatic embryo-derived plants significantly produce the highest number of suckers with an average of 21 suckers per plant compared to sucker-derived plants with only 13. These suckers can be used by farmers as source of planting materials for the next cropping season and as source of additional income. As to yield, the somatic embryo-derived plants produced bigger fruits and the highest harvest per hectare. And in terms of fruit size distribution, the somatic embryo-derived plants produced the highest percentage of large to medium fruit size, while the sucker-derived plants were more of the small to medium-sized fruits. So in terms of net income, the somatic embryo-derived plants obtained the highest income, followed by the multiple shoot induction, and the lowest is the sucker-derived plants. Using the somatic embryo-derived seedlings as source of planting materials can give farmers higher net income of almost 200% more than the traditional sucker. So these are the pictures of the fruits produced from somatic embryo-derived plants or plantlets and those from sucker. So this ends my presentation and maraming salamat.
Thank you so much for that very interesting presentation, Dr. Moreno. Those are certainly another two interesting biotechnology applications in agriculture and aquatic sectors. Our farmers and fisher folks will benefit from the outputs of these undertakings. We have received a lot of questions for our last two speakers, and the floor is now ready for the second set of Q&A. But before that, let me call again Dr. Nanola and Dr. Moreno to join me up in the screen.
Okay, thank you, GN.
Our first question is for Dr. Nanola, and the question is from Ms. Marilyn Lopez. Sir, can you tell us more about the current status of reef fish diversity in the Philippines?
Okay, to be able to answer that, let us go back historically, just to give you a perspective. There was a paper by Carpenter and Springer in 2005. They produced a paper on the diversity of marine fauna, particularly reef fishes, in the Philippines, data recorded way back in 1908. And in that paper, they were able to establish that the highest diversity of shore fishes is in the Visayas Sea, or the internal seas that I described earlier. Now, I had this opportunity to be able to revisit many areas in the country, and then I did a similar paper to them And we found out that the diversity is no longer, the highest diversity is no longer in the Visayas area, but in the Sulu Sea, as well as the West Philippine Sea. In short, what I am telling is that there is already be a shifting of the species diversity within the Philippines in the past 100 years. So from Visayas region shifting to the Celebes to the Sulu Sea region. So the question on the current status of the reef fish diversity in the Philippines, we actually decline in terms of number of species, and some of them have local extinction. What we call is the extirpation. So there are several papers looking on that, and one of the on the records that one of the endangered species in the Philippines is the mameng or the Caelinus undulatus. Thank you, Jen.
Thank you, Dr. Nanola. Our next question is from Mr. Jonathan Andrade, and the question is addressed to Ma'am Luz. Ma'am, he would like to know the difference of the technology in comparison to the traditional method of producing pineapple seedlings or planting materials.
Okay, thank you for that interesting question. Now, using this somatic embryogenesis technology, we can produce a number of planting materials. So from one crown shoot tip, we can produce around 8,000 to 10,000 in a shorter period of time. That is around 12 months compared to the traditional method, which is suckers, which we can get in the field after 21 to 22 months from planting. So in just one plant, under a traditional method, we can only have 10 to 15 suckers. So using this somatic embryogenesis, we can generate or we can produce thousands of planting materials. And the most important factor that we have to consider in here is that the planting materials that we are producing through this technology are disease-free, uniform, and true to type. Thank you.
Thank you, Dr. Moreno. Our next question is from Mr. Reggie De Jesus. And he wants to know what would be the effect of the identification of FMAs or fisheries management areas in your current study. Can you share your insights, Dr. Nanola?
Thank you, JN. So in relation to the FMAs, for those who haven't heard of this is actually the fisheries management area that was declared by Bureau of Fisheries and Aquatic Resources. Now, in the earlier presentation, what we're showing is the taxonomy by doing the traditional as well as the contemporary approaches. And our next step actually, which we're going to do in our research proposal on Abraham, we're going to do population connectivity studies. So in population connectivity studies, we will determine which areas are connected or a bit disjunct from other bodies of water in the Philippines. So in that way, our study can actually support the fisheries management areas in looking at the exploitation levels in terms of the catch rates of these different areas for better management. So we are supporting other efforts of the government. Thank you, Jayan.
Thank you, Dr. Nanola. Our next question is from Ms. Girly Ann Capili, and it is addressed to Dr. Moreno. Ma'am, she would like to know about the advantages or benefits of the technology to our pineapple farmers if they will use the planting materials produced through this technology.
Okay, thank you very much, Girly Ann, for your question. Now, using planting materials derived from somatic embryogenesis technology, as I have discussed earlier, the planting materials that are derived from a somatic embryo are already well established, meaning they have a good root system. So the plants had a good start at the beginning since they have the well-developed root system, and they grow faster and produce more number of suckers. So based on the result of our field evaluation, the plants derived from somatic embryos are or produce bigger fruits, and this mean higher income for the farmers. Now, in addition to the higher income derived from the from plants rather derived from somatic embryos or from planting materials derived from somatic embryos, farmers can also get additional source of income from the suckers produced from the plants derived from somatic embryo planting material. So in my presentation earlier, the somatic embryo derived plants produced more number of suckers compared to the traditional suckers. Thank you.
Thank you, Ma'am Luz. Our next question is from Miss Melly Tenorio. Dr. Nanola, she would like to know about other technologies, or if you have any technology to preserve our reef fishes. Sir?
Thank you, Mary, for that question. So the way I understand the question is that she wanted to know if we have this technology to not the preserve in can or reef fishes, but eventually let it what they call it propagate. So as far as I'm concerned, we have technologies in relation for the species that we farm, like tilapia, bangos, etc., and so on. But for the reef fishes per se, there have been technologies developed how to culture them also. But for the other reef fishes that have play a major roles in our ecosystem, we do not have that. But what we can do actually is basically protect our coastal environment, not only the coral reefs, but as well as the seagrass beds and mangroves, because these habitats are actually a part of the life cycle of the reef fishes. So I hope I gave you what we call it, a broader picture of what should we do or what should be doing in terms of the preserving or protection our reef fishes. Thank you, Jayan.
Thank you, sir. Okay, we are now on to the last question. The question is from Mr. Albert Malinaw Padilla. Ma'am Luz, ang tanong po niya ay paano nakatutulong ang inyong pag-aaral sa pagpapalaki ng pinya at kung may mabuting epekto daw po ba ito sa ating kalikasan? Ma'am Luz?
Okay, so thank you for your question, Albert. Now, I hope I got your question right, no? So basically, this somatic embryogenesis technology is more on the production of or on the rapid mass production of quality planting materials, in this case, specifically for queen pineapple. Now, this technology, okay, as I have shown earlier, the planting materials that were produced using this technology, okay, has already an advantage in terms of having a well-developed root system. So pag itananim natin ito sa field, so it means that the plants are already or has the capability of early establishment and utilizing the nutrients that are found in the soil. So they have already the advantage when it comes to faster growth and development compared to the traditional planting materials, which are the suckers na wala pang masyadong mga roots, no? So the sucker planting materials still have to develop roots para sila maka-establish and then that's the time na they can well established, or then they can already absorb more nutrients and grow and develop compared to doon sa somatic embryo-derived plantlets na mayroon na siyang magandang root system. Now, in terms of its effect into the effect sa environment, parang so far marang wala naman siguro ano doon sa environment, no? Kasi these are just the same as the traditional suckers when grown in the field, meaning they are both exposed to all the environmental factors in the field. So I hope I have answered your questions. Thank you.
Thank you, Dr. Luz. I hope our speakers have answered most of your questions. Let us hear once more from our speakers for their short message. You may go first, Dr. Nanola, followed by Dr. Moreno right after.
Thank you everyone for listening. So my message is that our country has the highest species diversity of species globally per unit area. So we should keep it that way by supporting efforts on marine conservation and let us not pollute our seas. Thank you.
Luz.
Okay, so thank you again, JM. So to all our listeners and participants to this webinar, so thank you very much for sharing your precious time with us. And again, I would like to emphasize that the use of good quality planting materials, which means that these are healthy, vigorous, uniform, and true to type. So this is the first important consideration for a successful and profitable farming enterprise. Now, the somatic embryogenesis technology offers a very good alternative in the mass production of quality planting materials, in this case for queen pineapple, so any time of the year, and this could significantly help boost the pineapple industry. And I am very optimistic that this technology on somatic embryogenesis is also applicable to other agricultural crops, especially those highly seasonal economic crops. Now, this technology can be extended to government agencies and institutions interested to help and assist our farmers in the distribution of good quality planting materials, especially in times of calamity and pandemic. So thank you.
Once again, thank you, Dr. Cleto L. Nanola Jr. and Dr. Luz Moreno. To our participants, if you wish to discuss further with our speakers, you may reach them through the email addresses showed up on the screen. We had 4 very interesting and informative topics on biotechnology applications for the improvement of the environment and economy. I hope our viewers have learned a lot from this webinar this morning. To give us a brief summary of all the information shared to us by our 4 experts on their respective fields, let me call on DOST Picard's Biotech focal person, Ms. Abigail May Oretuta. Ms. Abby?
Thank you, JN. So for this morning, we had a very fruitful discussion on 4 research initiatives of DOST Picard, highlighting the importance and use of biotechnology as tools to develop products, protocols, and technology to further improve agricultural and aquatic productivity. Our partner agencies shared with us the following. Biotech UPLB developed the Nutrio Biofertilizer, proven effective, and is now being commercialized for the improved productivity of sugarcane. Nutrio is a bacteria-based foliar biofertilizer which promotes nitrogen fixation and acts as a supplement to chemical fertilizer. Pilot testing in selected sites showed benefits such as soil pollution and chemical runoffs mitigation, long-term land sustainability, increased yield and income by 10 to 20%, while reducing cost of production in sugarcane. On the other hand, IBS-UPLB studied bacteriophages as it shows promising potentials as biological control agent against bacterial soft rot of vegetables. This post-harvest disease has always been a perennial problem and effective control measures is yet to be developed. In addition to the survey and collection of soft rot bacteria samples, an efficient isolation and identification pipeline was developed for soft rot bacteria, while tissue assays were done to determine their host range and pathogenicity. Moreover, lytic bacteriophages infecting Pectobacterium species were isolated, purified, and characterized. Finally, in vitro challenge tests using single suspensions and cocktail of lytic phages resulted in bacterial inactivation. The application of molecular methods through DNA barcoding was exemplified in the research initiative of UP Mindanao in the assessment of the biodiversity of reef fish species in northern and southern Philippines. The team have recorded over 150 market fishes in northern Luzon and 373 in southern Mindanao. Interestingly, the island chain of Basilan, Sulu, and Tawi-Tawi contributes most to this number with over 304 unique fish species. Altogether, 200 genetic barcodes are currently generated, and this will be made available in a biodiversity database also being developed. The speaker underscored that understanding the marine resource diversity through the integrative taxonomic approach combined with the traditional and contemporary molecular technologies is fundamental in any conservation and management efforts. Finally, through a series of experiments, VSU developed a protocol for rapid mass production of queen pineapple through the Somatic Embryogenesis technology, hence providing a year-round available supply of planting materials. The resultant tissue cultured planting materials have well-developed root and shoot system, more vigorous and disease-free, thus making them excellent source of planting materials. Results of field evaluation of the different types of queen pineapple planting materials show that somatic embryo-derived tissue cultured planting materials perform the best in terms of number of leaves produced and suckers produced, fruit weight, yield, and net income. This implies that somatic embryo-derived tissue culture planting materials are highly suitable as source of planting materials as they can survive under similar growing conditions as with suckers and have comparable agronomic performance. This may prove beneficial to the pineapple industry as availability of quality and disease-free planting materials has shown to be a major limiting factor in the productivity of the said crop. We hope that through these interesting topics, we cultivated awareness, deeper understanding, and appreciation on the practical applications and benefits of biotechnology. Thank you, and back to you, Jayen.
Thank you, Ms. Abby. I would just like to remind our participants to accomplish our CSF form to be entitled for the electronic certificate of participation of today's webinar. You may notify us in the email address showed on your screen if you will not receive a copy of your e-certificate after 10 working days. To formally close our program, we have this morning Dr. Edna A. Anit, the Director of the Crops Research Division.
Good morning, everyone. Thank you for joining us in this year's SNT Agri-Aqua webinar on biotech applications. The technologies that were presented by our resource speakers this morning are among the front runners in advancing the application of biotechnology in the field of agriculture and aquatic resources. Evidently, biotechnology has provided contributions and benefits on environmental sustainability and economic stability. Thus, Picard continues to bank on biotechnology to close the gap on agricultural production and provide sustainable agriculture. True enough, our researchers continues to prove effective and have been providing greater impact in our society. We are very thankful for the efforts, continued support, and unending passion of the project teams in delivering the expected outputs of their researches. In particular, we are very thankful for the guidance and leadership of the experts from the National Institute of Molecular Biology and Biotechnology, Biotech, and the Institute of Biological Sciences of the University of the Philippines, Los Baños, the University of the Philippines, Mindanao, and the Visayas State University. Our experts not only gave us good discussion on the accomplishments of the projects, but on the hindsight, they also help us better understand and appreciate biotechnology, its use and application on environment and economic improvement. Rest assured that DOS Picard will continue to support the responsible use of biotechnology as a tool to strengthen the three pillars of sustainability: social sustainability, environmental sustainability, and economic stability. We will be looking forward to more advancements brought by agricultural biotechnology, and will support our experts so that together we will have an effective and efficient agriculture sector. In closing, I would like to thank you all for attending this webinar. I hope that you found this activity very useful and interesting. Likewise, I hope that you continue joining the week-long celebration of the 16th National Biotechnology Week. Make sure to like and follow the NBW page on Facebook to learn more about the exciting activities and webinars about the latest in biotechnology in the country. You may also visit the virtual exhibit at www.nbw.gov.ph. Again, good morning and mabuhay tayong lahat.